My name is Eric Peterson. I am the co-director of digital recruitment and scholarship and associate professor of psychiatry and behavioral sciences here at USC. So during the talk today, if you have any questions, please use the Q&A feature. You can also use the chat. That's totally fine too. I'm going to monitor both the Q&A and the chat. So as questions come up, you can put them in there. Our speaker will likely field those questions towards the end, but if there are some pressing questions, we might be able to answer those during the talk. So we are really excited today to have Dr. Lana Ruvolo Grasser. She's going to be talking about the selection and utilization of wearables for biosensing in clinical, laboratory, and naturalistic settings. Dr. Grasser is a postdoctoral research fellow with the Neuroscience and Novel Therapeutics Unit within the Emotion and Development branch at the National Institute of Mental Health. And at NIMH, she's using neuroimaging and psychophysiological measures to study irritability, anxiety, and how they can be used in treatment for youth. She received her BS from Michigan State University and her PhD from Wayne State University. And she received funding from NIMH for her dissertation project looking at risk and resilience to trauma in Syrian refugee youth that were exposed to civilian war trauma and then forced migration. She received the 2022 International Society for Developmental Psychobiology Dissertation Award for this work. Dr. Grasser has also extended this work and continued working to address trauma-related psychopathology in families resettled as refugees of Syria, Iraq, the Congo, and Afghanistan. And she's led efforts to extend these programs to the virtual space for school children and to neighborhoods across Detroit for youth and caregivers. She's also passionate about science policy and advocacy and is a member of the National Science Policy Network and a recipient of the American College of Neuropsychopharmacology and the Americans for Medical Progress Fellowship in Biomedical Research Awareness. And so we are very excited to have Dr. Grasser here today, and I'm going to pass it over to her.
Thank you so much, Eric. Thank you so much for having me here today, and Karen as well. Thank you for all of your help in getting this organized. Very excited to be here with you all today. So as we said, we're going to be talking today about the use of wearables for biosensing across various settings that you may encounter in your research or in your clinical practice. Just by a quick way of disclosures, my funding and other organizations that I partner with, just to note that my presentation doesn't reflect the views of the National Institutes of Health, nor do I speak on behalf of the NIH. I also want to highlight that while I'm going to talk about wearable devices for biosensing, I will not be talking about specific names or naming devices during this talk, as the intention is not to tell you what device to use, but rather to give you a set of guidelines and helpful selection criteria for picking a biosensor that might be best for you. And through my introduction, you may have heard that I'm someone who, in my graduate work, worked really closely with persons who have been forcibly displaced. So it's always important for me to provide a land acknowledgement at the beginning of my talks in order to recognize and respect Indigenous peoples as the traditional stewards of the land that we reside and work on, and also to encourage us to support efforts to give land back. So in Detroit, where I'm from and where a lot of my graduate research was conducted, we are sitting on the lands of the Musqueam, Assassina, the Peoria, the Miami, the Mississauga, the Anishinaabe, and the Bodwodweepwin. And where I live currently and work in present-day Bethesda, Maryland, we are on the land of the Pescataway and the Notchtank. So I think this quote is a nice guiding principle for the why behind doing this work with wearable biosensing and measuring psychophysiological correlates of human health and behavior, especially when we're working in the mental health space. That's the space from which I approach this work in considering indicators of what youth might be at risk or resilient to a psychiatric disorder and how we can optimize treatment for those youth. So in this paper, they state to be measured with high validity, psychopathology must be observed within the dynamic contexts in which symptoms manifest. So this might be encouraging us to go beyond a traditional research assessment where we're asking someone to perhaps report on symptoms or tell us about their experiences when they're in a lab. The context might be a little bit different from their everyday life. They might not be experiencing a current stressor, or if we're having even folks do a task in the lab, it might not necessarily be as evocative of experiences that they have out in the real world. And again, maybe when they're dealing with other daily life stressors. So when we're able to have methods that we can use in both the laboratory setting, our clinical setting, and naturalistic settings, we can get closer perhaps to the validity of those constructs that we're interested in related to psychopathology. And we can also very likely enhance the inclusivity of the science that we're doing, not just restricting and requiring folks to come into our labs or clinics in which we might have geographic, transportation, timing, work-related barriers, but we can also have opportunities to measure, again, in those naturalistic contexts, in participants' homes, while they're at work, while they're at school, out and about living their daily lives. So we remove some of those structural barriers to participating in research. So here's just a brief outline for our plan today. We'll first start talking about the why behind why we might even be interested in psychophysiological data collection and using wearable biosensing. I'll then provide some considerations for selecting a wearable biosensor. So what do we want to think about when guiding our choices there? And then I'll talk a little bit about how we can optimize data collection and our data analytic pipelines. So let's first start with the why. And I want to just get a sense of where everybody might be coming to this space from. So you can go to slido.com, and if you just type in that number there, you should be able to join the presentation, join the conversation, and respond to some polling questions. Because as we talk about the why, I want to make sure that this is in fact oriented towards the why that some of you might have by getting a better sense, again, of where you're coming to this presentation from. All right, so my first question for you all, again, if you go to that Slido, is to know a little bit about what perspective you are coming from today. So again, if you missed the code, it's here on this screen. And then I'll flip over so that that question can become active for you again. So if you just want to put in here from what perspective you're coming to this as today. So it looks like the majority of us are coming here today from a research perspective or a clinician researcher perspective. That's great. And a few clinicians in the room as well. Thanks, everyone. All right, for the next question, I'll also just get a little bit of a sense of your familiarity, of what your familiarity with wearable biosensing is. So it sounds like we have some overlap. I also come to this space as a researcher. It sounds like the majority of you come to that space with that shared identity. And I'll disclose here that I'm someone who uses biosensing in research. I'm not someone who develops these types of tools, but I utilize them in these settings. So I see that we've got a fairly even distribution here from having no familiarity with biosensing at all in any of our research practice or personal use, having some curiosity in integrating that, and maybe having already used it in a personal capacity, and then some of us who share that experience in using biosensing in their research. Okay, that's great. Thank you all for sharing that with me. So circling back to our orienting question of the why would we consider physiology or psychophysiology as a unit of analysis? Well, if we look at the RDoC dimensions, the research domain criteria, we see that physiology is one of these units of analysis through which we can better understand things like valence, cognitive functioning, social processing, arousal, and sensory motor processes within and across different environments and contexts, and across time over the course of development or maybe pre, post, and intervention. And I think physiology is a really readily available unit of analysis because we can get at some of these mechanistic questions going backwards in this diagram towards those circuits, for example. But these are readily measurable substrates. We can measure physiological processes using non-invasive wearable biosensing, and we can do it not just when someone's in a lab and we're able to take a blood draw from them, for example, but we can even do it, when somebody is at home and just, again, in a naturalistic context. Now, I mentioned to you that I come from this space from a mental health background. And so this is really how I think about the information that we get from a physiological measure mapping on to a psychological construct that we might be interested in. So we know that physiological responses can be an index of a representational structure, and that might be a representational structure of something that reminds us of a scary, stressful, dangerous, traumatic event, reminds us of something that we fear or something that may signal danger to us. It might be something that makes us anxious, worried, or frustrated. And so if we measure the physiology when that representational structure is activated, whether it's through actually experiencing the thing that makes us scared or angry or frustrated, or perhaps receiving associated sensory information that can evoke the same physiological processes, we're able to then measure the physiological processes that underlie the associated behavior. So if we're getting really angry, mad, upset, we might be able to measure a change in the physiological state of an individual that drives or is driven by or is associated with that behavior and maps onto that representational structure. And if we take this kind of into the cognitive behavioral space, focusing on those responses that we have, which do include the physiological, we know that our behaviors are linked to our thoughts, are linked to our physical feelings and emotions. And so physiological signals then have a place in the assessment of cognitive, social, and functional domains. For example, we know that cognitive behavioral therapy helps us to learn how to become aware of our autonomic thoughts, how they're related to our autonomic behavior, and how they're related to our physiological processes, so that we can build an awareness of the connection between the behavioral, the physiological, and the cognitive thought. So again, if we're really interested, in better understanding, again, my perspective coming from a mental health space, who might be at risk for psychopathology, who might be resilient, and who is going to respond best to what treatments so that we can optimize the medicine and the therapies that we deliver, then we know that we have to consider a whole host of contributing factors that are going to influence why, for example, one treatment is not as effective as another treatment for someone else, or again, tell us why, even though two people might have the same lived experiences, one of them goes on to develop psychopathology and one of them does not. And again, the neurobiological and physiological state of an individual has been shown to be associated with treatment response and to be important in indicating us about precluding factors that might predict risk, resilience, and response. And these physiological variables, these same variables can not only tell us, what treatment might be best for somebody else, but they could also tell us how somebody is doing during the treatment. So we can use these indicators to measure whether or not somebody is actually responding to the treatment. A great example is exposure-based therapies. Are we seeing, for example, the increases in arousal when we expose somebody to, say, a feared stimulus? Or do we not see a response, right? We need to activate that representational structure in order for exposures to be effective. And then finally, we can use these physiological variables as a way towards long-term evaluation. So in addition to clinician ratings of improvement, self-report of symptoms, can we see whether or not we've actually made a change at the physiological level in terms of influencing how somebody is doing through a therapeutic process? So that's the why. Why would we be interested in measuring psychophysiological data and using wearable biosensing? So now I want to talk about the what in terms of actually picking a biosensor. So of course, our first mission is to identify what are our constructs of interest and what units of analysis do we want to look at. And I mentioned two main domains that we might be interested in looking at, especially in the mental health space. So one is arousal and reactivity, and some physiological indicators of arousal and reactivity include skin conductance level and skin conductance response, both of which are measured through electrodermal activity, and they are indicators that are derived from that measure. We can also look at startle response. This is a muscular response. If we're doing a more lab-based paradigm, that can be made to be a mobile paradigm and done across multiple settings, we could look more specifically at fear-potentiated startle to something like threat cues. We can look at arousal, or heart rate, a very canonical measure of arousal. Temperature. And sometimes temperature can also kind of be a corroborator of the data that we derive from heart rate or skin conductance to ensure that we're getting a good signal and a proper measurement of physiological arousal. We can even look at activity. And then on the other end of the spectrum is regulation. So again, we can use some lab-based measures to look at regulation. And then we can also look at things like heart rate variability, which is a great indicator for us of emotion regulation, flexibility of systems to adapt to change. So these are just some of the potential constructs of interest that you might be looking to use in the clinical or the research space, all of which can be measured using a number of different types of biosensors, devices, and systems. So then there's developing and or actually choosing that wearable. And based on some of your responses to the poll that I first shared, it sounds like most of us are in the clinical validation space, where we actually want to use these tools in our research or in our clinical practice. But of course, before that is the verification and the analytic validation stage. So when choosing a wearable, if you're not in that development state, it's really important to have an encompassing literature review that you're sure that there is supporting evidence that the sensors you're using are accurately capturing the data that you're interested in, and of course, outputting data within expected physiological ranges. And then that the algorithms that are used to derive different metrics of whatever unit of analysis you're interested in are functioning properly, and that these metrics are stable and accurate. So just to give an example of this, there are a lot of wearable devices that are wrist-worn wearable devices that might look something like a traditional watch. And many of those devices have photoplethysmography sensors, or PPG, using light to measure the changes in blood volume pulse, from which we can derive measures of heart rate, an indicator of arousal, and heart rate variability, an indicator of regulation. Now, the gold standard for measuring heart rate and heart rate variability in medical settings would be something like an EKG or an ECG, electrocardiogram. And a lot of times in past research studies, that's done with a series of electrodes that are placed on the body, or it may be done with a chest band. Again, that's worn just above the diaphragm. And so in many of the analytic validation studies for devices that use PPG as opposed to ECG, they've compared and contrasted their data for the PPG sensor compared to a gold standard, well-validated ECG sensor. And that way we're able to have an understanding that we're capturing the data that we want, everything is functioning properly, the resulting metrics are stable and accurate. So those are the things that you might be looking for in the literature when choosing your wearable device. Some devices for their sensors also have FDA clearance. Just because a device has FDA clearance for one of their sensors, some devices might have multiple, doesn't mean it's for the device as a whole or for all sensors. So that's also something to look out for. So in the next series of criteria to be thinking through when selecting a wearable, we're going to really focus in on this clinical validation end of things. So there's kind of four main things that I have here that you might consider very broadly when selecting a wearable device. The first being the duration of the assessment that you're looking forward to doing. Do you want to just make an assessment during a task that somebody is doing perhaps on a mobile device or computer at home, or on a task that they're performing in the lab, or while they may respond to some questions, say in a clinical setting? Or do you want the individual to wear the device, to utilize the device over an extended period of time, perhaps days, weeks, even months? So if you're going to be doing continuous sensing, of course, you need to think about whether or not the sensors are actually sampling data continuously. So on some devices, the sensors turn off at night in order to conserve battery, or might not routinely sample throughout the day. They might turn on and off intermittently to make recordings, or be driven by user input to turn on the sensor and make a recording, again, to conserve battery life. So that's one consideration. You also need to consider the sampling rate. If you're using the device during, say, a task that, again, you're administering on a mobile device, computer, or maybe you're doing it in lab or clinic, if the task has a very short time course, perhaps on the order of seconds or milliseconds, then you need to ensure that the sampling rate for the physiological data you're interested in is on a similar order. If you're getting summed data that maybe is aggregated over a 1 minute time period, that might not be enough information, sensitive enough if you are having an event related design where things are changing on the order of seconds that you're interested in. You have to consider, of course, the cost. That's definitely always going to be a rate limiting factor. And if you're going to need to get multiple devices so that multiple people could wear them for days, weeks, months, over an extended period of time, say in your research study, that's going to incur an extra cost. And these devices range widely in their costs. Usually the devices that are oriented towards research are more expensive than the devices that are oriented towards individual consumers. And you also have to think about data storage. This kind of goes into battery life as well and continuous sensing. But are data going to be stored locally on the device? Is the user going to have to somehow upload their data at certain time points? Or is that data going to be uploaded to a cloud? If it is, does the user have access to Wi-Fi in order for those connections to be made and for data to get uploaded, or do they not? And is there a potential risk of data being overwritten because the device can't handle any more storage without pushing an upload to the cloud? And you also have to think about security. Is the device and its data associated platforms HIPAA compliant, for example, if you're doing this work in a clinical context? You then have to think about the units of analyses that you're interested in. So are you interested in just one psychophysiological indicator? Maybe you're really interested in heart rate or heart rate variability. So having a PPG sensor or an ECG is all you need. Or are you interested in, say, PPG and skin conductance response that you're deriving from an electrodermal activity? Well, then you need two sensors now. Maybe you're also interested in temperature. Maybe you're also interested in movement. So you need an accelerometer and a gyroscope. So you have to consider these units of analysis and then understand whether you have one device that has multiple sensors. So many of the watches, for example, have multiple sensors built in. Or do you have a system that might have multiple devices? So perhaps there's some platform that you're working with where they have different sensors that all need to get connected, or perhaps on their platform, they offer an array of biosensing, but you can only do one at a time with their setup. So all considerations. Next is location. Are you going to be collecting these data in the lab, in the clinic, or in a more naturalistic setting? In the lab and the clinic, it's probably more readily accessible for you to be able to charge the device and not have to worry about the battery life and also distributing chargers to your participants. And you might have the opportunity to set up the device and all of its components for the participants. So for example, if you're thinking back to a system that maybe can measure heart rate and skin conductance simultaneously, then perhaps that's not a problem when you're in the lab and you can set up all of the different electrodes across all of the different parts of the body, and you have the entire system there ready to collect data. Maybe in the clinic, this becomes a little bit harder because you don't necessarily have the time to go through this complex setup if you have a lot of patients who are coming in. But maybe if you're interested in something like heart rate or heart rate variability, because you're in a clinical setting, it might be feasible to have somebody put on a chest strap and to be able to make sure that that's getting worn correctly. But perhaps if you're working in a more naturalistic setting, say that you're working with kids and you want to do assessments while they're in their naturalistic settings at home, it might not be as easy for the family to set up a chest band, for example, and make sure that there's good connectivity and that the kiddo is comfortable wearing it throughout their day. But it might be more user-friendly if they have a wrist-worn device that they can simply put on like they would wear a watch, or many kids even have Apple Watches these days and are familiar with that. And then finally, data integration. So first and foremost, one of the biggest questions is whether or not you, the researcher, have access to the raw data before anything is done to it. So many different companies that distribute wearable devices may have built-in proprietary algorithms. Sometimes you can get a lot of information on the back end about what exactly is being done. Perhaps it's just some basic cleaning, noise reduction of your data. But perhaps on the other end of the spectrum, you're just getting summary data. You're getting these calculated variables. You don't know how the data was cleaned or processed. You don't know how the summary variables were calculated. And maybe again, if you're interested in looking at data at specific times, say that you pair this with EMA and you want to look at, physiological state 20 minutes before at the time or 20 minutes after the time of the beat. And perhaps if the time course data, that summary data is given to you from the device is not on par with where you're trying to sample, you might miss out on those actual data epochs that you're most interested in. And also think about the capabilities of the device in pairing with various software development kits. So for example, if you're interested in performing wearable biosensing, maybe in a naturalistic setting, and again, going back to that example with EMA, are you able to actually pair the device that you're using with the EMA platform? And of course, this can be very beneficial for, again, tying perhaps some responses that participants are giving about their psychological state with their physiological state. And many researchers are also working in this state in the space of just-in-time adaptive interventions. So are you able to get a physiological measurement from the device that can then pair with some sort of intervention facing the participant, something that may ping them through, again, a mobile app, for example, that says it's time for you to take a few deep breaths. It looks like you're getting a little bit stressed. And not all wearable devices actually have the capability of pairing with other applications. So this is another consideration, of course, to think about. So I want to give one more specific example when we're thinking about the units of analyses and the capabilities of biosensors. And I'm going to talk a little bit more specifically about heart rate variability. And the reason for this is because heart rate variability is considered to be one of the best indicators in terms of if we're interested in looking at constructs like stress and anxiety and understanding an individual's regulatory capacity. So this might be of interest for many folks. It's an area that's of interest to me. And heart rate variability as an indicator, I think, really speaks to the complexity of the considerations that need to be made when using wearable biosensing in clinical and research spaces. So you have the idea, okay, I want to look at heart rate variability. I know that's important for mental health. First question is when? Are you just looking at heart rate variability at rest or at a baseline period? So maybe prior to doing a task, prior to having a clinical conversation, maybe talking about a traumatic experience or going through some sort of clinical intake or interview forms? Or are you talking about heart rate variability during rest in a naturalistic setting? Or are we interested in reactivity? So how does heart rate variability change perhaps when we introduce a stressor in a lab context, or perhaps when you report maybe through EMA that you experienced a stressor today, you were feeling pretty stressed out? So the timing, again, is going to be important, and that taps back into what type of data you're getting from your biosensor. Do you have access to the raw data? And if not, what epochs are data binned into so that you can be sure you're getting the right timing of the data that you're interested in looking at, because these are really dynamic processes. Then you want to consider, again, what you're interested in and the time course. So there are many different indicators of heart rate variability that map onto different physiological processes and regulatory processes. So there are components in the time varying domain, and you see those listed here on the left side of the screen. And the components from the time varying domain are considered to be reliable even for shorter periods of time, so 5 minutes or less, for example. And these time varying components can pretty reliably be measured using either photoplethysmography, which is again the sensor that a lot of the wrist-worn devices use, or using ECG, which of course would be a setup using electrodes, using a chest band, or in some cases, using a sampled on a wrist-worn device, but that usually requires a timed sample and is not taken continuously because it's using a special sensor on the device. If you are interested in these frequency domain components, a longer sampling duration is typically required. And because of the signal, the reliability of the signal that you get and the type of signal that you're measuring, using ECG is typically recommended. So the PPG signal is again measuring the blood volume pulse, and that signal looks a little bit different in terms of its waveform and the information that you can derive from it compared to the waveform and the information that you can derive from ECG. And because of that, we typically consider ECG to be a better indicator when you're interested in these variables from the frequency domain that you can see on the right. And I'm not speaking so specifically so that we don't get too much into the weeds, and because there's some debate in the literature, But all of these 6 different indicator variables are considered to map onto slightly different physiological processes. And so again, being aware of what the kind of higher order construct you're really interested in is going to be important for guiding your selection. And you can see how there are so many different considerations just for this one physiological indicator to take into account. So I'm going to zoom back out, and on the next slide, I'm going to show you a chart that's kind of checking off maybe some criteria in terms of the sensors that you might be interested in looking at, and then we'll add some additional information. So I'm showing to you here 8 different devices and systems that are currently available. Again, I have not named these because my intention is not to aim you toward a specific product or away from one or the other, but really just to show you, again, the options and things that you might consider. So you can see here where a check mark appears. It means that device or system has sensor capabilities for measuring ECG, PPG, EDA, temperature, or activity through accelerometers and gyroscopes. And so you can see many of the systems, for example, have a measure for photoplethysmography or PPG from which you can derive heart rate and heart rate variability. Many also have an accelerometer and a gyroscope. Of course, this is really helpful then when we look at our physiological data as well, which we'll talk about to be able to take motion into account for artifact that we might observe in our data. You'll see a little less common is electrodermal activity, that measure of sweat on the skin, from which we can derive variables like skin conductance level, so the tonic component, or skin conductance response, the phasic component that we would typically lock to some sort of event or to stimulus. And in my own personal capacity and research, I personally think it's great to have a device or a system that also has EDA if you're interested in looking at heart rate and heart rate variability. For example, if your data is messy from the PPG signal, say, or if you have other barriers that have somehow caused artifact in your PPG data, then you might have on hand EDA as an alternative indicator. So it can be nice to have multiple potential indicators if something fails. Temperature also is a good one just to ensure that you have contact with the skin, and many pipelines for data analysis use temperature as an inclusion-exclusion variable just to ensure that data are within physiological ranges and you actually had good contact with the skin. So now on the left side of the screen, I've added some icons to tell you a little bit more about what the different setups might look like. So many of the devices in wearable biosensing are wrist-worn devices. These are really common, especially for their user-friendliness. Many of us might use a wearable biosensor like these in our real lives to monitor our own exercise patterns or maybe our sleep patterns, or just to help us enhance our communication throughout the day. Some of these systems, the ones that are marked systems, might use electrodes and different sensors, whether they be wired or wireless, to provide additional capabilities. And then one device that's listed on here is a ring that would just be worn on the fingers. So again, when you're thinking about these devices, you have to think about the user friendliness. Of course, if you're getting a wrist-worn device and you're working with children, for example, or a ring device, you have to think about the size and whether or not it's going to fit them. Now, I've highlighted the system B and device C just to note that these are examples of some that have actually been developed first and foremost for research purposes. Many of the wearable biosensors that folks might use in research clinical space may not have necessarily been developed for research as the primary intention, or may now in their current utilization and current development might be more geared towards consumers for personal use. So just to highlight the intention of what this biosensor that you're interested in using might be, something that's developed for research might have better access to raw data for researchers, right? Because of that intention in the development. But it might not be as user-friendly if you're aiming to use the biosensor in a naturalistic setting for individuals. I also highlighted System G because there are some out there that, the user friendliness of it might be a little bit more complicated, but they have been developed with a user in mind. So just to highlight that there kind of are some of these in-between stages as well. So I used a little cool looking emoji to highlight some of the ones that, again, might be more user friendly because of their development. One of these systems that is a little bit more tricky, but has guides at least that guides a user in terms of the setup. And then one of the systems that really does not give the user any information about how to do the setup and requires a more in-depth technical knowledge in order to set up that system. So something like this might be more appropriate for a clinical or a lab-based setting. And then finally, I wanted to note System G. One of these systems is an example of a system where, even though it has the capabilities to target different physiological targets, you cannot simultaneously collect data. And so that might be a barrier to research if you're interested in looking at heart rate and sweat at the same time, for example. All right. So we've talked a lot about considerations that, and I just highlighted here again, I think I said this earlier, but I just wanted to highlight here again that heart rate and heart rate variability in the field of stress and anxiety in particular, and in health and mental health more broadly, are considered really reliable and valid indicators of arousal, of regulation, and of overall health. So again, these might be variables that are of great interest to someone, and you just need to prioritize when selecting your biosensor. What are the goals of your research or clinical practice? All right, so we've talked a lot about the what, and now once you've selected your biosensor, how can you optimize your data collection and think about your analytic pipeline? All right. So first, in terms of data collection, having good training is really the offset for being able to collect good data. Whether you're in a clinical, a lab, or a naturalistic setting, good training is going to result in higher quality data. So in the lab or in the clinical setting, I always like to employ a do one, see one, teach one, or really three, but it doesn't sound as nice, strategy for teaching. So if you're first able to have somebody observe you, perhaps doing the setup on an individual, especially if you're working with one of those systems that's a bit more complicated setup of sensors or electrodes, then you have the individual actually do it themselves while you're observing and giving feedback. And finally, if they can teach somebody else how to do the setup, that really shows mastery of the skill and in-depth knowledge. If you're working with individuals who are going to be using a biosensor at home, maybe you're sending someone home with a wrist-worn device, for example, sending them home with both a written manual and a video demonstration, right? These are 3D things that we're working with. So sometimes having an actual visual of how to put the device on correctly can be helpful, and being really specific in the information that you provide. So if you're able to tell someone, here are where the sensors are that we're collecting data from, that can help the individual recognize how to set up the device properly, knowing that, okay, this is the part that needs to be touching my skin, and giving a bit of a greater understanding. When it comes to the setup itself, making sure to clean the skin appropriately prior to the application. So that might mean having the individual wash their hands and their wrists thoroughly, Washing with a soap that's non-alcoholic based can be really important if you're interested in measuring something like electrodermal activity. If you use hand sanitizer or if you use an alcohol-based sanitizing soap, that can actually dry out the skin. And so it's going to deplete the signal that you're able to measure, and it's going to confound the measurement that you're making of the sweat on an individual's skin. So good to wash the hands and wrists with soap and water, maybe non-alcohol based. If you are doing something like measuring, for example, ECG or another type of sensor that you're using on the skin, in those cases, you can certainly use a little alcohol wipe as well to ensure that the skin is nice and clean and there's no debris that's interfering. Speaking of that interference, if it's possible and necessary to remove any hair at the point of contact. So again, perhaps you're wearing a wrist-worn device, and maybe the individual has a lot of hair that might be interfering with where those sensors are placed. So being able to remove any hair at the point of contact, if that's a possibility. Ensuring good contact with the skin without excess pressure, right? So if you're putting on a wrist-worn device or you're fitting a ring, whatever it might be, ensuring that there's actually good contact with the skin, that it's not slipping around, jiggling around on the wrist. And again, that kind of feeds into reducing movement of the sensor against the skin. Sometimes when individuals are working with an electrode, they might tape down the ends of the leads that are coming out of the electrode, again, to reduce any movement that might be occurring there. Reducing any ambient light interference. So again, that kind of all goes back to having good contact with the skin. If you have a wearable on the wrist, for example, and there's a gap in between and you can kind of see those LED lights that are being used for photoplasmography, you might end up having some ambient light interference as well, as you probably don't have good contact to even be making your measurement. And then, of course, reducing any other interfering signals. This is probably going to be more relevant if you were doing something like using a multi-electrode wireless system in a lab or clinical-based setting, but ensuring that you're reducing other inference. Perhaps you don't want the person to be doing their task with their setup with a phone, also because it could just be distracting. And we know that these devices are so pervasive, so making sure to remove that interference. And if someone has a watch, maybe having them remove whatever bracelets or anything else that they're wearing on the wrist from that arm. So my quick and easy tip for most of the points that are listed here, those that have to do with getting good contact with the skin essentially, goes back to the 80s and is really all about wristbands. So if you put a sweatband on over a wristband, this is what we actually do in one of our clinical trials. We have kiddos who are wearing a wrist-worn device while they complete exposure-based cognitive behavioral therapy. And we put a little sweatband over the device. One, it's fun because they get to pick out a sweatband in their color that they get to keep, and that's exciting for them. But two, it helps ensure that we keep good contact with the skin, we reduce any ambient light interference, and it's not jiggling around on their wrist, especially because they're kiddos and they're a little smaller. And then finally, if you can obtain any corroborating or alternative data in case one of your sensors fails, for example, it can be helpful to have some sort of a backup. Like I mentioned, if you're interested in measuring arousal, maybe you're measuring heart rate and electrodermal activity. In terms of developing an analytic pipeline, there's a lot of things to think about here. First might be software. So, you have to understand what processing has already taken place by the company itself. You might be interested in using free and open source software, and I can tell you that there are free and open source softwares for processing all of these data types that I've covered. There's a lot of marketing, of course, towards the paid and licensed versions, but there all are alternatives. How you will handle missing data or artifacts. Sometimes, again, this is built into proprietary algorithms of a device, so being aware of whether you need to repeat or add that step or not in your analytic pipeline. Artifacts can arise, technical artifacts due to things like missing extra or misaligned beats, for example, with PPG or ECG, measurement noise or inaccuracy of a detection algorithm. And there's also physiological artifacts like for heart rate and heart rate variability, again, ectopic beats and arrhythmic events. And then having your process for how you're going to handle noise and motion, right? If the individual is moving around, for example, can you apply a filter? Are you going to remove data segments that are really noisy? Or maybe if that's related to a construct of interest, which it very well could be, are you going to use motion as a covariate? And then, of course, what outcome variables are you going to select? Like with HRV, there's many different variables that you could have. And this is just an example, again, with electrodermal activity, the phasic component being skin conductance response, the tonic being skin conductance level. And again, those many choices you have for something like heart rate variability. And then finally, in your pipeline, you might be interested in linking this with other data types. So is it locked to a task and you want to link events together? Or is it locked with something that's occurring in the clinic? For example, like I said, my work, we collect data during CBT. And so we want to time lock different physiological events with when they're doing the exposure, when they're debriefing from the exposure, et cetera. And then finally, for your analytic pipeline, your considerations, are you going to pre-register hypotheses? Certainly, you're probably collecting a wealth of data, so being transparent and specific about what hypotheses you had at the offset and what exploratory questions may have been prompted. A lot of this data, especially things like heart rate variability data, can be very non-normal, and so you might need to apply a log transformation or use a parametric test. You're likely collecting high density data, so things like multi-level models might be best suited to handle nested structures across time. And you may need to use nonlinear modeling and, of course, two-tailed tests to capture the realities of these types of data. So there might be a range that is a physiologically normative response for a stressor, like the line plotted in green. Your heart rate should go up, right, when you encounter a stressor. But folks who have that exaggerated response might be more at risk for psychopathology. And so too may folks who have a blunted response. And we actually know that in terms of cardiovascular disease risk, those who are on both extremes, highly reactive or highly underreactive, are at greater risk. So being able to model that accurately in your data. And we know that there are so many other factors that contribute to risk resilience and response to treatment. So we want to be modeling things like the participant's age, their sex, as well as their race. And I want to highlight here that race can encompass a number of other unmeasured variables. In one of my colleague, Dr. Nathaniel Harnett's work, he found that race was a proxy for experiences of discrimination that had altered the physiological tone of individuals. And so accommodating for that in the models was very important. Now, based on some of the work that I do, here are some kind of practical applications of how you might use this data. In the work that I do, we're interested in whether arousal and regulation as measured by these psychophysiological indicators predicts response to cognitive behavioral therapy, whether we see habituation within sessions. So if I present you with something that makes you really frustrated or angry, can I activate that system, but also bring it back down throughout the session? And then finally, over the course of sessions, are we able to change your overall levels of arousal, maybe bringing that down and improving regulation? So again, multiple contexts and settings that you can use wearable biosensing in, just sharing a few examples from my work. This is a more canonical lab-based paradigm of fear potentiated startle in youth that we've found to be an indicator of youth at risk for post-traumatic stress disorder. Thinking of something that we can use in more dynamic contexts, our skin conductance response to trauma interview protocol. We use a mobile application with just a set of sensors that are very low cost and easy to set up. And we can measure this biomarker reliably in homes, clinics, and in labs and get the same pattern of results. I mentioned to you also the importance of considering racial and ethnic differences here that might be driven by variables like exposure to stress and discrimination. We've used these methods to look at augmented reality exposure therapy, and actually showing here that augmented feared objects drove the same physiological response as the real feared object. And then finally, the study that I talked about that's currently ongoing at NIH, where we're looking at using wearable biosensing during cognitive behavioral therapy to look at changes within and across sessions. So I'll stop here to take questions and to really acknowledge all of the people who have contributed to the work that I'm doing currently at NIH and our collaborators. I will selfishly mention that I'm starting at Wayne State as a faculty position in the fall, and will be taking graduate students for 2025. So if that's something that you might be looking forward to, perhaps you'll consider applying. And then I'm happy to take questions at this time.
Great. Thanks, Dr. Grosser. This was fantastic. We learned a ton. I know I definitely learned a ton. So thanks for walking us through. Yeah, let's see if any questions come up as people want to get those into the chat or into the Q&A. I just had one that I was thinking of when you were showing that grid of the systems and the devices and the green check marks. I wonder, when you were looking into this, did you find that that any of the devices or systems, like I know you had, I think, one or two that covered all the areas. I mean, did you find that they were really made to cover like one of the areas really well, and the one or two that covered all of them maybe just kind of spread themselves too thin and weren't really good at all of them?
That is a great question. So the one that's indicated there that does cover every indicator that I had listed was built for research purposes. And so it does do all of those components well. It is the system that's the most difficult to set up, the most complicated. It's very costly. So it's not really, I would say, easy to implement in many settings unless that is your area of research, because of that cost and the time that's consumed by the intensive analyses that need to be performed and things like that. In terms of a lot of the wrist-worn wearables, many of them were developed to monitor sleep and to monitor exercise and overall health. So they seem to do the best with looking at things like heart rate using photoplethysmography to look at sleep. So that might be their accelerometers and gyroscopes, as well as their temperature sensors, and again, their heart rate sensor that do well with those. And in terms of heart rate variability, I think that is more of a limitation of the type of sensor, the type of measurement, photoplethysmography writ large. That becomes a bit of a barrier for wearable devices and isn't necessarily as specific to a device, because that signal it can map reliably onto ECG signal, but there are still some discrepancies there, especially when you're looking at components in the frequency domain. And I think that's a more pervasive issue for PPG in general, rather than specific to a device.
Well, very cool. Oh, thank you. Okay, any other questions before we wrap up? I know we're at time here, so if anybody has any questions, you can always send them to me as well as to Dr. Grosser. Yeah, this was really great. We really appreciate your time and you coming and giving this fantastic talk. Everybody, if you can see, there is a evaluation link that Karen put into the chat. If you could click on that, just take you a minute, let us know what you'd like to see in the future. And we'll send those over to you, Dr. Grosser, when we get those.
Sounds great.
All right, well, thank you very much. We really appreciate you and your time. And yeah, we will see everybody. I guess we will be back in June. So keep your eyes open for an email from us advertising what we're going to talk about next.